Yeah, okay. All right, thanks. Is that better? Okay. All right. The first groups of slides I'm going to go through fairly quickly because I've shown them before, and I want to include them because there are some new people here, but so they have been reviewed for some of you maybe. I'm going to finish up with brown marmorated stink bug and spotted-winged Drosophila, and those are really two of the hot topics that everybody needs to hear about. So if you have questions on the borers and the thrips, I'll be glad to answer them. And my email address will be at the end of the program as well, so you can contact me individually. All right, so I'll be talking about borers, thrips, and invasives. Oh my. All right. Just seeing if you're paying attention. All right, the first one of the borers is raspberry crown borer. This is a clearwing moth in the same family as peach tree borer, grapefruit borer, dogwood borer. Most of these are wasp mimics, and as you can see, this one is a pretty good mimic of yellow jackets. The eggs are laid in the summertime on the undersides of leaves. These hatch and after about a week and a half, and they'll hatch in late July, and they will drop down to the young larvae will go down to the crown, and they'll overwinter there in a hibernaculum at the base of the plant. In the spring, it will tunnel into the plant and into the cambium, and will feed in that area. And you may see a reddish-brown sawdust-like frass coming from cracks in the bark in April. In the second summer, it moves up into the stalks and tunnels in the stalks and can cause them to break. Now, in the early studies, which were done in the northern part of the range, there was a 2-year life cycle reported, but more recent work in Arkansas shows in the southern part a 1-year life cycle. Now, the material, there were a couple of materials that were used for years as a drench, diazinon and azanphosmethyl. Those are gone now. Those are not available for use. 7 or malathion can be applied as a foliar spray, and bifenthrin, a pyrethroid insecticide, can be used as well. Now the label says 200 gallons per acre in this drench application, but research in Arkansas showed that you don't need that much volume. 50 gallons per acre work just as well. And this should be applied between October and early April, either at in the fall, after the young larvae are down there, or in the spring before they have a chance to tunnel into the cambium. Now, for organic growers, entomopathogenic nematodes have been used. Generally, in crops where entomopathogenic nematodes are used, nematodes that are beneficial that feed just on insects, one thing that's needed is adequate soil moisture. They can't tolerate standing water, but they need moisture in the soil, so irrigation helps a lot in the success of this approach. Cultural control, infested canes and crowns should be removed from your planting. Now this last comment on the screen applies to all 3 of the borers. Get rid of wild brambles nearby, and this will reduce the infestation pressure in your cultivated planting. Now, the one I've been talking about, raspberry crown borer, it may be hard to diagnose. It seems to me that it's the least common of the 3 borers, but there's a hidden problem there in that the insect is hidden. It's cryptic, and many infestations probably get by undiagnosed. But potentially it's the most severe of the borers, more likely to actually kill the plant. Now, the other 2 borers are very common. You've probably seen the damage from these insects, the symptoms. The first one is rednecked cane borer. This is a buprestid beetle, one of the metallic wood boring beetle families. This makes a elongate gall on the stem of the plant. You see that widened area. This is a very common symptom. I worked with a berry grower in Southwest Virginia several years ago, and almost all of his plants had these swellings from the larval infestation of the canes. This is about 1/4 of an inch long, and the pronotum, which is a section right behind the head, is coppery color, iridescent and coppery color. That's very characteristic. And you can see these beetles on a sunny day resting on the plant. The adults are present from May to August, depending on the region, maybe a little later. The females lay white eggs on the trunk. The larvae exit directly into the trunk, so they're never exposed to sprays. So what the target, when you spray for it, you're targeting the females laying eggs, not the larvae as they're tunneling in, because they're not exposed. And here we see some illustrations here. Once they're inside, they spiral around the cane. You can see on the right where the larvae have caused a girdling of the plant. As you can imagine, this really disrupts the translocation of the plant, and you can get loss of vigor of the plant or death of that part of the plant. So these reach about 12 millimeters long, and you really don't see the larvae unless you're digging in the plant for them. They're internal feeders. The larvae winter here, and in the following spring, they pupate in the plant. And then they're about 20 to 40 days, about a month in the pupal stage. Now, you may see D-shaped holes on the plant. This is the emergence hole. When the adult leaves the pupal chamber, it makes a D-shaped hole. This is characteristic of the whole family. So if you have birch trees, you may have bronze birch borer. It makes this kind of hole. Emerald ash borer makes a D-shaped hole on ash trees. This is a family characteristic. But this is the only one in raspberry and blackberry, so you know what pest it is. Now, after leaf fall, if more than 10% of the primicanes are infested, then you can decide the spray is justified. Examine primicanes for adults during that time when the adults are active twice a week, beginning at bloom. And then you can spray either malathion or brigade, an OP or a pyrethroid, at intervals 7 to 12 days apart, as while the adults are present. Remember, it's the adults laying eggs that are the target of the sprays. And again, wild host removal is very helpful. That grower that I mentioned in Southwest Virginia, he did use malathion, but he also removed the wild brambles that were nearby the planting. And within a year, he eliminated a major infestation to almost nothing. Okay. Now the last is raspberry cane borer. This is a longhorn beetle, a cerambycid. And you see it has very long antennae, unlike the last one. It's about twice as long, about ½ an inch long. Now the pronotum is colored too. It's kind of a red-orange color with 2 black spots. It's not the coppery iridescent color that you saw in the redneck cane cane borer. So the adults appear in June, and the female, when she's laying eggs, makes 2 rows of punctures. 2 rings of punctures are near the tip of the shoot. I'm sure you've seen the symptom if you've been growing blackberries or raspberries. That's from this insect, and the shoot tips will wilt in the early summer. Now, and what will happen is the larvae will continue on down past that area if you let it away, if you don't remove those injured tips. Wilting canes, the wilted areas can be removed. And if you do this within a few days after it starts to wilt, you can remove the larvae that way before they have a chance to tunnel down into the plant. Malathion here works as well as insecticidal soap, MP, which is an OMRI-approved material. So that's it on the borers. Again, I'll answer questions, but I'll move on right now to thrips. Thrips are a growing concern, not just on caneberry crops in Virginia, but on many crops worldwide. They're really becoming a global issue. One of the main pests is western flower thrips. Now we have, well, you might call it eastern flower thrips or just plain flower thrips. That's native to the east. Western flower thrips used to be a western species, but it began moving 10 or 20 years ago, moved through North America, and now it's in Europe as well. And this is a key player because it's a much more effective vector of viruses than the eastern flower thrips or any of the other thrips that live on caneberries as well. The Thrips Life Cycle. You see the eggs are laid on the plant. The first two stages, the nymphs, immature stages, these are free living on the plant, feeding on the plant. The next two stages are the pre-pupa and a pupal stage. These are sort of a quiescent stage. They live in the soil. And then when the adult stages reach that, the adult leaves the soil and returns to the plant, feeds, and lays eggs. The females lay eggs. Now, there's a wide range from year to year in intensity of thrips populations. Part of this is whether or not it's a very dry year or high amounts of rainfall. If there's a lot of rainfall at the right time, it can drown the soil stages, the pre-pupa and the pupa. There's more natural mortality in very rainy years. In years where it's real droughty, there's very low natural mortality, and you have more thrips infestation on the plant. Another issue is that thrips are one of the groups of pests that have been seen to be flared by pyrethroid applications. Pyrethroids are inexpensive to use. They're relatively safe for human applicators, but they're very damaging to natural enemy populations. And so that we sometimes see outbreaks of thrips where pyrethroids are used. So if you're going to use pyrethroids, you should be aware of that and be watchful for these pests as well. By the way, one of the more effective predators of thrips seems to be aureus, the little minute carrot bug that's fairly common during the summer. That's a generalist feeder. It feeds on eggs of many pests, mites, aphids, and thrips. Now, the impact of thrips, you know, when you talk with entomologists, you'll hear different descriptions of the symptoms of thrips feeding. And it seems, from my observations, I think, you know, when you see a single white druplet on a berry, I think that might be due to thrips feeding. We need to do more cage studies, but with something as small as a thrips, it's hard to do, you know, cage studies. But I'm not talking about when you have the whole side of the berry bleached out. That's more related to sun injury. But when you have the individual white berries. Another thing that one of my graduate students noticed, who was making counts in raspberries for other pests, was that sometimes when she pulled off the raspberry from the receptacle, there were a lot of thrips feeding inside, between the receptacle and the berry, which I think is something that most people haven't been aware of. Role as virus vectors. Thrips do transmit several viruses, and especially the western flower thrips. Now, in the mid-Atlantic states, these viruses have shown up in caneberries. The real relationship to disease is unclear, because many of these viruses don't really cause much symptom unless the virus is occurring concurrently with another virus. And so it's a very complex situation. So we've seen several viruses transmitted by thrips in the caneberries, but you know, we're not, the actual disease effect is not clear yet. But consumer effects, if you have a container of berries in the marketplace and there are thrips crawling across the surface, that's one thing that consumers do notice. And you know, that can be another marketing issue. There's always the human factor. All right, insecticides for thrips. Now, one thing to note, there's a list of materials here. Some of these are OMRI certified, some are not. But whether or not you're conventional or organic, you have to worry about insecticide resistance. And thrips are a group that have been known to develop resistance widely in various parts of the world. So if you find an effective material, you can't keep hammering the thrips with that same material, because you'll drive it into the ground. You need to rotate among different mode of action classes, and sometimes we don't have that many to choose from. So we have Azadirect, which is a neem product, Azadiractin, that's OMRI certified. We have Entrust, which is OMRI certified, and that's really all on this list. Now, if you're conventional and you can use Delegate, that's the same mode of action class as Entrust, so that would not constitute a rotation. You know, it has to be a different mode of action class. So on this list we have neonicotinoids, we have spinosins like Delegate and Entrust, and organophosphate, and then a botanical, the Azadiractin. So these are several materials to apply, and Admire Pro can be soil applied as well. Now, The hot topics. We just had the survey of brown marmorated stink bug, and we see that you see an adult and a nymph here, and this is an import from China. It showed up in mid-Pennsylvania, Allentown area around 1996. It was first found in Virginia in 2004. It took several years for it to build up to damaging numbers. After a few years, it reached high enough populations where it would come into houses and create domestic infestations. And then after it reached that level of abundance, it continued to grow, and it reached a point, especially in 2010, where it was common enough that it was causing agricultural injury in the field during the summer. And it really hit the newspapers in 2010. So here you see the adult and the nymph. One character that really separates it easily from the native stink bugs, for example our brown stink bug, which is not very closely related, it's a different genus, is a brown marmorated stink bug has white bands on the antennae and also on the legs, although the bands on the legs are not always as prominent. On the nymph on the right, you certainly see the distinct bands on the legs. They're a little more diffuse in the adults, but those white bands on the antennae are very characteristic. So the number of generations has been unclear. We're getting a little bit more information on that now, and we'll continue in 2012. When it was first found in Pennsylvania and New Jersey, it was reported to have one generation. Now it looks like in northern Virginia, which really is not that far from Pennsylvania and New Jersey, there may just barely be time to have two generations in the season. It's a real matter of concern in the southeast because, do you have a question?
Are they related to the Asian longhorn beetle?
No, it's another of the Asian pests. The question was, is this related to Asian longhorn beetle? This is in the Hemiptera, it's a true bug. Asian longhorn beetle is in the same family as that raspberry cane borer that I showed earlier with a real long antennae, but that's a beetle, and that's easy to tell apart. We have a series of pests from China in recent years. I'll try and come back to that at the end of the talk. This is a new one. We aren't clear on some of the aspects, even of the basic biology. In southern China, it can have up to 6 generations. And so how that relates to its new range as it spreads south down into the deep south, we're not certain yet in the terms of the numbers of generations. In China, it feeds on a wide range of plants. It has more than 300 different host plants, which is much broader than the native stink bugs. The native stink bugs are prolificous too, but not apparently as prolificous as this one. In the fall, it seeks shelter in houses. And so why the big stink? One is literally because of the stink. One is it has such a wide host range and it attacks so many different kinds of crops. Now the yellow states are where it has been found. It's spread after its initial introduction into Pennsylvania. The states in red are where it has caused the most injury so far. And we're definitely in that range. Here we see apples that have been fed on. I took those pictures up in Maryland at a grower meeting at Catoctin Mountain. Those apples are really unsaleable. That corking under the skin. In fact, just driving into that block, it looked like the trees have been hit by a hailstorm. It was so conspicuous with that denting and the discoloration on the fruit. On the lower photos, that was a vineyard where we were doing an insecticide trial. In 2010, there was a concern among wine grape growers because the first runs of harvested berries, they were crushing stink bugs with the grapes and causing a tainted juice. And so that's a big thing into the wine industry. So we did an emergency spray trial where we used belay, which is a neonicotinoid, and we used pyrethrin, pyganic, a botanical insecticide, just the evening before harvest, before the crew was going to be in there. Both of those materials have a 0-day PHI on grapes. And both worked at getting the bugs out of the clusters. The pyrethrin possibly even a little bit better than the synthetic. So we have that answer, but that may be a moot point because Joe Fiola at the University of Maryland has been doing some work with crushing known numbers of bugs in lugs of berries and and seeing how few it takes for a panel to be able to detect the taint. But once it, well, there's an earlier concept called ladybug taint. That comes from Harmonia, but that's a different talk. But what Joe is finding, that after the wine has fermented and aged in the bottle, then it was no longer detectable. So it may not be, that may not be an issue. There's still an issue with the beetles damaging the berries. All right, now let's look at what the eggs look like. Now, one of the issues of uncertainty is, well, on many of the tree fruit crops, the native brown stink bug uses those crops as a feeding host, not a reproductive host. So the adults will go feed on the fruit, cause some injury, and then leave. And they're reproducing on weeds and soybean, herbaceous hosts like that. A big problem with this stink bug is this one uses trees and vines as a reproductive host. So the eggs are laid on the plant, and then all the nymphal instars, the whole life stage, are feeding on the fruit. So that is one of the reasons why this is a worse pest than the other stink bugs. Why it's not just another stink bug in the mix. Now let's look at the eggs. The eggs are usually laid in batches of about 28. And the native stink bugs may vary in the numbers. And there's some variation here too, but it's about 28 usually. They're barrel-shaped eggs with kind of a round lid on the top. And I'm going to try and use the arrow here, the cursor, to point these out. There are 3 screens, and I can't point to all of them. You see around here, there are little projections. Those are the horns that both the egg breathes through. Oxygen enters there, and sperm enter after mating to fertilize the egg. This is very characteristic of stink bug eggs, to have this row of horns. But the degree of development varies among stink bugs. And I want to show one particular one. The one on the left, a couple times last year, fruit growers showed me these egg masses and asked if they were the egg masses of brown marmorated stink bug, and they're not. Look at the size of the horns. They're very elaborate. It looks like a starburst, and the eggs are gold colored. This is another stink bug, but this is a beneficial stink bug, Podesis. This is a predatory member of the family. So these are actually beneficial. So you have to look close, and not all stink bug eggs are the same. And we're trying to develop a pictorial key to the eggs of these different stink bug species. Right now, these pictures will be online. Okay, see if this works. Well, it's not really working. This is a video clip. I want to talk a little bit about the injury, but the video is not going to work. This is a video that my former graduate student, Laura Maxey, took. This is a green stink bug feeding on a raspberry. And the same behavior was shown by green and brown stink bugs. And the stylets are inserted between the drupelets of the berries. And you could see, with a video clip, you could see working it down in, pulling out a little bit, then working back in. And this is late berry development feeding, and I'll come back to that in a second. But what this leads to is you see collapsed drupelets, and in this one you can see the puncture holes on all 4 sides of the druplet, and this can lead to rots and further breakdown of the berry. Now, if the feeding happens earlier, what you may see is the whole blossom being killed, and this is early season before bloom, and this can be a big problem. And I heard one estimate of a grower losing 50% of his berries last year because of brown marmorated stink bug. So we'll be continuing to look at the effect during the season in our planting at Kentland Farm this season. Now, the materials of choice are going to differ on the fruit crop. You have to check which berry crop or which fruit crop you're growing. Now, there was a list of materials that was developed after lab studies at USDA in early 2011, and some of those didn't necessarily work well once you took it to the field. So we need to do field testing of these materials. One material that rated very highly on that list was malathion. Now malathion is still registered on caneberries and grapes. It's not registered on the tree fruits, most of the tree fruits anymore. But this one was fairly effective on brown marmorated stink bug. Some of the neonicotinoids are effective as well. Acara, Assail. The pyrethroids like Brigade or bifenthrin, Brigade or Sniper, are effective. Now, one thing you have to be careful of, as I said, the pyrethroids often flare populations of secondary pests. And a particular one, I'll be talking about this at a meeting next weekend, in vineyards. If you use pyrethroids to control various pests, grape berry moth or whatever, one pest that can be flared is grape mealybug. Mealybugs are the vector of grape leaf roll virus. And so, right now, we don't have pyrethroids at all after early season in our vineyard pest management guide because of this concern. As we'll learn to deal with brown marmorated, we might have to reconsider that. But right now, we don't have pyrethroids in the vineyard guide. We do have them in the berry pest management guide because we don't have leaf roll virus. But this issue of flaring secondary pests and how they fit into an IPM program is a continuing area of research for us. By the way, we had two botanicals there on the bottom of the list. We have pyganic, which worked at killing them, knocking them out of the plant, but you wouldn't expect pyrethrum to have a very long-term effect. It would be a very short residual material. A newer product came on, was approved by EPA this past year, Azera, which is a mix of pyrethrins and azadirachtin, the neem product. So I haven't seen that used yet in the field, but I plan on using it in the coming season. Now, just in case you're getting bored with just one new invasive pest, we have a newer invasive pest to keep things interesting. Anybody that's been growing fruit has run into Drosophila at some time or other. The little, what the public calls fruit flies that breed in overripe fruit, like overripe bananas and things like that. There's a new one that just entered Virginia last year, midsummer in 2011, spotted wing Drosophila. This is very different than the other Drosophila species. Drosophila suzukii, another Asian species. We're not sure exactly where it came from. I'll show you a map of the range in a minute. Here we see the eggs in strawberry. Now you can see these with the hand lens. You know how big a Drosophila is, so you can imagine the eggs are very small. But you see these white eggs, and you can see a pair of white horns here. Those are respiratory horns. That's how these eggs breathe. They're little spiracles, little openings on the ends of these horns. Now, you might think these are microscopic since the fly is so small, but these are next to the achene of a strawberry. You know how big the achenes of a strawberry are, and so you can probably see these fairly easily with a hand lens. It'd be hard to see it with the naked eye. And here we see the young larvae in the strawberry. These will cause the beginning of breakdown in the fruit and a premature softening and beginning of rot in the berries. Now the way you identify them is the male has a spot on the wing. Now you have to look close and take note of where exactly the spot is, because some of the native Drosophila that don't cause damage have some spots on the wing. There's a fairly common one that has 2 spots on each wing, but this one has a prominent dark spot in the leading edge near the tip of the wing, and so that's very characteristic of the males. And there's also some dark stripes on the front legs, but this is very hard to see even with a hand lens. I wouldn't bother looking for that. Now the female is different. The female lacks the spots, so you're going to have to look at her hind end for the ovipositor. Now, this is the ovipositor on a regular Drosophila, like Drosophila melanogaster, one of the native ones. I can get the arrow back here. There we go. You can see barely some small teeth along this structure. That's a very small ovipositor. It can't penetrate intact, healthy fruit. That's why it only lays eggs in overripe fruit. Compare that with the ovipositor of spotted wing Drosophila. It's a big, hardened tooth structure. And this can penetrate the skin of fruit, especially the soft-fruited, soft-skinned fruit. And again, for comparison. Now, this one is a very photogenic individual on the female on the right. The ovipositor is normally that produced, that extended. Normally, this structure is tucked up underneath here, like this one would be. But you could still see, if you look with magnification, you could still see that dark brown area under here with the teeth. It's not going to be stuck out like that. And you can see this especially if you have magnification, a hand lens, or a dissecting scope nearby. Now, here's the life cycle. The overwinter as adults. The life cycle is 20 to 30 days. Although it can complete a generation from from egg to egg in as little as 12 days. That will depend a lot on temperature. The eggs hatch in 12 to 72 hours, half a day to 3 days, depending on the temperature. You see the little larvae, no head capsule, just a typical little maggot developing in the fruit. Now the history, this is widespread in southern Asia, from Japan all the way to northern India, so through the bulk of China as well. We don't know exactly where this came from. Now, it was introduced into California in 2008. In 2009, it moved up through California, Oregon, Washington State, into British Columbia on various fruit crops. Caneberries, strawberries, blueberries, berries, grapes, and cherries as well. Also late in 2009, Florida. And so we knew that it would be heading our way. So in 2010, we took part in a trapping study for this species. Now the traps, it's a very simple trap to make. You can do this at home. The bait is just apple cider vinegar. And use plastic deli cups with little holes punctured up on the top. You don't want these holes to be too big because you want to keep out earwigs and things like that. Now this is cloudy on the left because this is one of our traps early at Kentland Farm. We were including yeast in here. We thought the yeast would increase the attraction to the Drosophila. It turns out it didn't really increase the attraction to the Drosophila, but it made it a lot more unpleasant to work with. It really smelled bad. And so on the right is just clear apple cider vinegar. We use sticky yellow trap cards in there, and you can do this if you want. You can buy these things for they're sold for greenhouse supplies, and then the flies will get stuck to that. Then you can take those out, bring them back to the lab, put saran wrap around it. But this year we're going to do away with the cards, and we're just going to pour the fluid through a filter and then into alcohol to bring back to the lab to look at. And you have to look at them close because you'll get a lot of native Drosophila in there as well, not just a spotted wing Drosophila. This is not a very specific trap. Now, in 2010, the flies were detected in South Carolina and North Carolina, right up to Virginia. We did not find it in Virginia in 2010, but we knew we could expect it very shortly. And so in 2011, we were trapping in 5 sites. The first site where it was found is where the traps were being checked very frequently in Hanover County, central Virginia. Then the second find was in Sussex County, and although this is far to the south, it probably got missed at first because this trap was being detected as part of being monitored as part of a different trapping program for other pests. These were traps located at the Sussex County landfill, and so there's no telling where those flies came from. We then found it at the research station up in Winchester, and then the research farm at Kentland Farm. And then finally, in the fall, in September, at Virginia Beach. So we found it in all 5 of the sites we trapped in Virginia, northern, southern part, eastern, and western. So we consider that it's entirely distributed in the state now. Oh, no way. Now, this map was generated at the end of the season. This was taken off the Exotic Pest Monitoring Service site online. The yellow and the purple mean the same thing. It's just different ways of reporting. This fly now has spread through Virginia, up through New Jersey, Pennsylvania, through New England. So really now it spread up through the west coast very quickly, and it spread along the east coast very quickly. So the host list. Really all the fruit crops grown in Virginia are on the host list. Also tomato, and we need to investigate this. It was reported from cherry tomato in Florida. We need to document this and see how much at risk they are. This could be a big problem on the eastern shore and in central Virginia, where tomatoes are an important crop. So cherries are a preferred early season host, grapes are a preferred season host, but the berry crops are at very high risk. I put together a list which is the best impression we have so far. This is really an educated guess. We don't have any real experience with it yet. But I think the crops that will be at greatest risk are the berry crops, the caneberries, strawberries, and blueberries, then cherries and grapes, then the stone fruits, and the pome fruits, and really the tomatoes are there or the question mark. We don't really know where they fall yet. So the eggs may be deposited on the surface, but that, if they're just on the surface, you can't tell if it's native Drosophila or Suzuki'i, the spotted wing Drosophila. If they're inserted in the tissue, it's almost certainly spotted wing, if it's otherwise intact fruit. Any fruit that's broken open can have Drosophila laying eggs. So here we see eggs on cherry, blackberry, blueberry, and strawberry. Then here you have a cherry with a larva that has come out. It punctures a hole, a bigger hole to leave. It will go down and may pupate. You may find the pupa in the berry, but often the pupae end up down in the soil where the life cycle is completed. So cultural control, harvest promptly. Now, although they can attack healthy fruit, they can also lay their eggs in overripe fruit like other Drosophila. So you want to prevent overripe fruit, harvest promptly to reduce breeding sites, destroy overripe or rotten fruit that are nearby. Now, this last recommendation is probably a moot point now. This one recommendation I found in another state where it had just been introduced on the West Coast, and they were recommending that if spotted wing is not in the area, and you find it, you should destroy the crop. Well, that's no longer effective. It's through the whole eastern states now. So I don't think you have to worry about destroying the crop if you find spotted wing Drosophila. Now on the bottom is just a web page on spotted wing I've set up in the Virginia Fruit website, and I'll show you another slide how to get there again at the end. For insecticides, this is easier to kill than brown marmorated stink bug, but the issue is it's at a time when you put the sprayer away and you don't want to be spraying anymore. It's right before harvest. And so you see several materials here, and these materials are in our small fruit and in our great pest management guide for this year now. Entrust and delegate, these are both spinosins, same mode of action. Malathion, which is an organophosphate. Mustang Max is a pyrethroid. Pyganic is a natural pyrethrin, a botanical material. Azera is a pyrethrin and azadirachtin mix. Now keep in mind that we have to worry about resistance with this. Resistance has already been reported in California, where pyrethrin, pyganic, has been applied repeatedly. With a number of generations, about 13 generations a year, and the number of eggs per female, there's a high risk of resistance in this pest. You have to rotate materials, and you have to, if you're going to grow berries commercially, you have to be watchful and be prepared to spray in late season. Now, entrust and delegate are the same mode of action, so that would not be a rotation. Mustang Max, a pyrethroid, and the natural pyrethrin are the same mode of action, and so that would not be a rotation. Now, Azera, which is pyrethrin and azadirachtin. The azadirachtin is a different mode of action, so that would constitute a rotation. So the materials that are in red here are synthetic. Conventional growers can use these, and these are three that are they're effective on spotted wing, and they're different modes of action. So these are three that can be rotated. Dept Delegate has a 7-day PHI, Malathion 3-day, and Mustang Max is 1 day. Now, if you're trying to be organic, we have 3 here that also have different modes of action. Entrust, which is a spinosyn material, Pyganic, which is the natural botanical, and azidiractin, which is also a botanical, but it works in a different way than pyrethrin. So those are some materials to start with. We hope to be doing some efficacy trials this year. And these materials are all in our pest management guides, which are online. Yes?
Which do you think is more effective, the non-organic or the organic?
Generally, I think the non-organic would have longer residual. And so they would probably be more effective in that regard. The pyrethrin is going to have a short life. In fact, that's why the pyrethroid class was developed, to answer the problem of the short life of pyrethrin. And so that's the way it is. So how many people here are trying to be organic? A lot of hands here. So so you would be limited to these, but be aware that the pyrethrum, you're probably already aware, the pyrethrum is going to be a very short residual material. Entrust would last longer. Azera, again, as I said before, on the brown marmoset, I don't have any first-hand experience with that one, but I think that will be longer lived because I think the azidiractin would last longer than the pyrethrin. Now, to find these sites, oh yes?
On the Malathion, find the process for home use. Would that also be effective?
I'm sorry, say that again?
Malathion, that process for home use, would that also be
Yes,
effective?
yeah, as long as it's the same amount of malathion, yeah, it's the same malathion in the home product as well as the commercial product, so that should work as well. Now, I showed the whole URL for some of the web pages for this, but you can find the web pages I've set up on these insects by just having in Google BMSB Virginia Tech or SWD Virginia Tech, and you'll come to the sites. You can go to the Virginia Fruit web page and work your way through the different crops and pest management and the list of pests. Yes?
If you had a crop and you wanted to destroy it because it was infested, how would you do that?
Well, that's a good question. One thing that's been tried was how to destroy a crop that's infested. Burying it right under the surface is probably not good enough because the mature larvae will still pupate on the soil and just escape through the soil. They often pupate in the soil. But one thing that seemed to work in preliminary trials was solarizing it, putting plastic over it and heating it up. But I don't really have any other recommendations on that. I haven't been involved with that research. Yeah, if you're going to bury it, you have to bury it pretty deep, I think.
If you just threw it in a river,
that's littering. There's another hand over here. Yes. Yeah, the question was permethrin. Yeah, permethrin, that should work. It's pretty broad spectrum, and you saw other pyrethroids on here too. The Mustang Max was one that was published, and that worked well. That's a pyrethroid. The pyrethroids generally have fairly good residual. Yes. So as I understand the question, is it better to use a mist sprayer or an air blast that comes from the side versus one that sprays from the top? Is that what you're asking?
Yeah, well, one of them has a lot more pressure, even if it's a lot more softer.
Well, I think if you're getting material onto the fruit and the clusters, I think that either one would work okay as long as you're getting coverage. Okay, so as we develop research proposals, I've just submitted one proposal on spotted wing. We're planning to submit another one very in the near future. If you've had issues with spotted wing, we could certainly use grower testimonials to add to our justification section of proposals. So that's my email address. Also, I have an online project management site for berry crops, a berry scholar site. It's by subscription basis. I enroll people by their email address, and I use this membership list as a basis for mailing for insect updates and things like that. And there's some information archived in here as well, like links to our spray guides, biological information on the pest. If you would like to be enrolled in that and you're not, send me your email address and I'll add you, and you'll start getting the updates as well. All right. So that's all I had. I'll be around till a break. Yes.
How come that the stink bug doesn't stink when you kill it?
How come they do or don't?
I don't smell it at all.
They don't always release the smell. You know, if you look with a hand lens, now I teach insect morphology too, and now you got to get a morphology lection. You know, the nymphs have these scent glands on the dorsal part, the abdomen. The adults, it's the ventral side of the thorax, and if you look close with a hand lens, you can see this groove between the middle and hind leg, and that's where they release the scent from. And maybe they've already released it all, and they don't have any more in the reservoir to emit. But one thing, you know, try this next time you're grocery shopping and see if you can smell it. One way I heard the smell being described is like cilantro. And so crush, next time you're in the grocery section, crush, don't tell the merchants I told you to do this, but crush a leaf and see if you can smell a little stink bug in there.
Yeah.
All right. And email me your, all right.